Class 11, one of the most important and basic chapter 8, Cell, the unit of life. When you look around, you see both living and non-living things. You must have wondered and asked yourself, what is it that makes an organism living, or what is it that an inanimate thing does not have which a living thing has? The answer to this is the presence of the basic unit of life, the cell, in all living organisms. All organisms are composed of cell. Some are composed of a single cell and are called unicellular organism, while others like composed of many cells called multicellular organism. Now, what is cell? Unicellular organisms are capable of first independent existence and second performing the essential functions of life. Anything less than a complete structure of cell does not ensure independent living. Hence, the cell is the fundamental structural and functional unit of all living organisms. Anton von Leeuwenhoek first saw and described a live cell. Robert Brown later discovered the nucleus, and the invention of the microscope and its improvement leading to the electron microscope revealed all the structural detail of the cell. Cell theory. In 1838, Matthias Schleiden, a German botanist, examined a large number of plants and observed that all the plants are composed of different kinds of cells which form the tissue of the plant. At about the same time, Theodore Swan, 1839, a British zoologist, studied different types of animal cells and reported that cells had a thin outer layer which is today known as the plasma membrane. He also concluded based on his studies on plant tissue that the presence of cell wall is a unique character of the plant cells. On the basis of this, Swan proposed the hypothesis that the bodies of animals and plants are composed of cells and product of cells. Schleiden and Swan together formulated the cell theory. This theory, however, did not explain as to how new cells are formed. Rudolf Virchow, 1855, the scientist first explained that cell divided the new cells and formed from the pre-existing cell, omnis cellula e cellula. He modified the hypothesis of Schleiden and Swan to give the cell theory a final shape. Cell theory, as understood today, is all living organisms are composed of cells and product of cells, and all cells arises from pre-existing cell. Now, an overview of cell. You have earlier observed cell in an onion peel or human cheek, cells under the microscope. Let us recollect their structures. The onion cells, which is a typical plant cell, has a distinct cell wall as its outer boundary, and just within it is the cell membrane. The cells of the human cheek have an outer membrane as the delimiting structures of the cell. Inside each cell is a dense membrane bound structure called nucleus. This nucleus contains the chromosome, which in turn contain the genetic material, DNA. Cells that have membrane-bound nuclei are called eukaryotic, whereas cells that lack a membrane-bound nucleus are prokaryotic. In both prokaryotic and eukaryotic cells, a semi-fluid matrix called cytoplasm occupies the volume of the cell. The cytoplasm is the main arena of cellular activities in both the plant and animal cell. Various chemical reactions occur in it to keep the cell in the living state. Besides the nucleus, the eukaryotic cell have outer membrane-bound distinct structures called organelle, like the endoplasmic reticulum, Golgi complex, lysosome, mitochondria, microbodies, and vacuoles. The prokaryotic cells lack such membrane-bound organelles. Ribosomes are non-membrane-bound organelles found in all cells, both eukaryotic as well as prokaryotic. Within the cell, ribosomes are found not only in the cytoplasm but also within the two organelles, chloroplast in plant and mitochondria and on rough ER. Animal cell contain another non-membrane bound organelle called centrosome, which help in cell division. Cells differ greatly in size, shapes and activities. For example, mycoplasmas, the smallest cell, are only 0.3 micrometer in length, while bacteria could be 3 to 5 micrometer. The largest isolated single cell is the egg of an ostrich. Among multicellular organisms, human red blood cells are about 7 micrometer in diameter. Nerve cells are some of the longest cells. Cells also vary greatly in their shapes. They may be disc-like, polygonal, columnar, cuboid, thread-like, or even irregular. The shape of the cell may vary with the function they perform. Now, prokaryotic cell. The prokaryotic cells are represented by bacteria, blue-green algae, mycoplasma, and PPLO, pleuropneumonia-like organism. They are generally smaller and multiply more rapidly than eukaryotic cells. They may vary greatly in shape and size. The four basic shapes of bacteria are Bacillus, Coccus, Vibrio, and Spirillum. The organization of the prokaryotic cell is fundamentally similar, even though prokaryotes exhibit a wide variety of shapes and functions. All prokaryotes have a cell wall surrounding the cell membrane except in mycoplasma. The fluid matrix filling the cell is the cytoplasm. There is no well-defined nucleus. The genetic material is basically naked, not enveloped by a nuclear membrane. In addition to the genomic DNA, the single chromosome circular DNA, many bacteria have a smaller circular DNA outside the genomic DNA. These smaller DNA are called plasmids. The plasmids DNA confers certain unique phenotypic characters to such bacteria. One such character is resistant to antibiotics. In higher classes, you will learn that this plasmids DNA is used to monitor bacterial transformation with foreign DNA. Nuclear membrane is found in eukaryotes. No organelle like the ones in the eukaryotes are found in prokaryotic cells except for ribosomes. Prokaryotes have something unique in the form of inclusions. A specialized differentiated form of cell membrane called mesosome is the characteristic of prokaryotes. They are essentially enfolding of cell membrane. Cell envelope and its modification. Most prokaryotic cells, particularly the bacterial cells, have a chemically complex cell envelope. The cell envelope consists of a tightly bound three-layered structures, that is the outermost glycocalyx, followed by the cell wall and then the plasma membrane, although each layer of the envelope performs distinct function. They act together as a single protective unit. Bacteria can be classified into two groups on the basis of the differences in their cell envelopes and the manner in which they respond to the staining procedure developed by Gram. Those that take up the Gram stain are Gram positive and those do not are called Gram negative bacteria. Glycocalyx differs in composition and thickness among the different bacteria. It could be loose sheath called slime layer in some, while in others it may be thick or tough called capsule. The cell wall determines the shape of the cell and provides the strong structural support to prevent the bacterium from the bursting or collapsing. The plasma membrane is selectively permeable in nature and interact with outside world. This membrane is similar structurally to that of the eukaryotes. A special membranous structure is the mesosome, which is formed by extension of plasma membrane into the cell. These extensions are in the form of vesicles, tubules, and lamella. They help in cell wall formation, DNA replication, and distribution to the daughter cells. They also help in respiration, secretion process to increase the surface area of the plasma membrane and enzymatic content. In some prokaryotes like cyanobacteria, there are other membranous extension into the cytoplasm called chromatophore, which contain pigments. Bacterial cells may be motile or non-motile. If motile, they have thin filamentous extension from their cell wall called flagella. Bacteria show a range in the number and arrangement of flagella. Bacterial flagellum is composed of three parts: filament, hook, and basal body. The filament is the longest portion and extends from the cell surface to the outside. Besides flagella, pili and fimbriae are also surface structures of the bacteria but do not play a role in motility. The pili are elongated tubular structures made of a special protein. The fimbriae are small bristle-like fibers sprouting out of the cell. In some bacteria, they are known to help attach the bacteria to rock in a stream and set to the host, also to the host tissue. Ribosomes and inclusion bodies. In prokaryotes, ribosomes are associated with the plasma membrane of the cells. They are about 15 nanometer by 20 nanometer in size and are made of two subunits 50S and 30S units, which when present together form 70S prokaryotic ribosomes. Ribosomes are the site of protein synthesis. Several ribosomes may attach to the single mRNA and form a chain called polyribosomes or polysomes. The ribosomes of a polysome translate the mRNA into protein. Inclusion bodies. Reserve material in prokaryotic cells are stored in the cytoplasm in the form of inclusion bodies. These are not bound by any membrane system and lie freely in the cytoplasm. Example: phosphate granules, cyanophyceans granules, and glycogen granules. Gas vacuoles are found in blue-green and purple and green photosynthetic bacteria. Now, eukaryotic cells. The eukaryotes include all the protists, plants, animals, and fungi. In eukaryotic cells, there is an extensive compartmentalization of cytoplasm through the presence of membrane-bound organelles. Eukaryotic cells possess an organized nucleus with a nuclear envelope. In addition, eukaryotic cells have a variety of complex locomotory and cytoskeleton structures. Their genetic material is organized into chromosomes. All eukaryotic cells are not identical. Plant and animal cells are different as the former possess cell walls, plastids, and a large central vacuoles which are absent in animal cells. On the other hand, animal cells have centrioles which are absent in almost all plant cells. Now let us look at the individual cell organelles to understand their structures and functions. Cell membrane. The detailed structures of the membrane was studied only after the advent of the electron microscope in the 1950s. Meanwhile, chemical studies on the cell membrane, especially in human red blood cells, enabled the scientists to deduce the possible structure of the plasma membrane. These studies showed that the cell membrane is mainly composed of lipids and proteins. The major lipids are phospholipids that are arranged in bilayer. Also, the lipids are arranged within the membrane with the polar head towards the outer side and the hydrophobic tails towards the inner part. This ensures that the non-polar tail of saturated hydrocarbons is protected from the aqueous environment. In addition to the phospholipids, membrane also contains cholesterol. Later biochemical investigation clearly revealed that the cell membranes also possess protein and carbohydrates. The ratio of the protein and lipid varies considerably in different cell types. In human being, membrane of the eukaryotic erythrocytes has approximately 52% protein and 40% lipids. Depending on the ease of the extraction, membrane proteins can be classified as integral and peripheral. Peripheral proteins lie on the surface of the membrane, while the integral proteins are partially or totally buried in the membrane. An improved model of the surface of the cell membrane was proposed by Singer and Nicholson in 1972, widely accepted as fluid mosaic model. According to this, the quasi-fluid nature of lipid enables lateral movement of the proteins within the overall bilayer. This ability to move within the membrane is measured as its fluidity. The fluid nature of the membrane is also important from the point of view of function like cell growth, formation of intercellular junctions, secretion, endocytosis, cell division, etc. One of the most important functions of the plasma membrane is the transport of molecules across it. The membrane is selectively permeable to some molecules present on either side of it. Many molecules can move briefly across the membrane without any requirement of energy, and this is called passive transport. Neutral solutes may move across the membrane by the process of simple diffusion along the concentration gradient, that is from higher concentration to lower concentration. Water may also move across the membrane from higher to lower concentration. Movement of water by diffusion is called osmosis. As the polar molecules cannot pass through the non-polar lipid bilayer, they require a carrier protein of the membrane to facilitate their transport across the membrane. A few ions or molecules are transported across the membrane against their concentration gradient, that is from lower to higher concentration. Such a transport is an energy dependent process in which ATP is utilized and is called active transport. Example, sodium and potassium pump. Now cell wall. As you may recall, a non-living rigid structure called the cell wall forms an outer covering for the plasma membrane or fungi and plants. Cell wall not only gives shape to the cell and protects the cell from mechanical damage and infection, but it also helps in cell-to-cell interaction and provides barrier to undesirable macromolecules. Algae have cell wall made up of cellulose, galactans, mannas, and minerals like calcium carbonate, while in other plant it consists of cellulose, hemicellulose, pectins, and proteins. The cell wall of a young plant cell, the primary wall is capable of growth, which gradually diminishes as the cell matures, and secondary wall is formed on the inner towards membrane side of the cell. The middle lamella is a layer mainly of calcium pectate which holds or glues the different neighboring cells together. The cell wall and middle lamella may be traversed by plasmodesmata which connect the cytoplasm of neighboring cells. Endomembrane system. While each of the membranous organelle is distinct in terms of its structure and function, many of these are considered together as the endomembrane system. Because of their functions are coordinated, the endomembrane system include endoplasmic reticulum, Golgi complex, lysosome, and vacuoles. Since the function of the mitochondria, chloroplast, and peroxisome are not coordinated with the above components, they are not considered as the part of the endomembrane system. The endoplasmic reticulum (ER). Electron microscope studies of eukaryotic cells reveals the presence of a network or reticulum of the tiny tubular structures scattered into the cytoplasm, that is called the endoplasmic reticulum (ER). Hence, ER divides the intercellular space into two distinct compartments, that is luminal inside ER and extraluminal cytoplasm compartment. And the ER often show ribosome attached to their outer surface. The endoplasmic reticulum bearing ribosomes on their surface is called rough endoplasmic reticulum. In the absence of the ribosomes, they appear smooth and are called a smooth endoplasmic reticulum. RER is frequently observed in the cells actively involved in the protein synthesis and secretion. They are extensive and continuous with the outer membrane of the nucleus. The smooth endoplasmic reticulum is the major site for the synthesis of lipid. In animal cells, lipid-like steroidal hormone are synthesized in SER. Golgi apparatus. Camillo Golgi 1898 first observed densely stained reticular structures near the nucleus. These were later named Golgi bodies after him. They consist of many flat disc-shaped sacs and cisternae of 0.5 micrometer to 1 micrometer in diameter. These are stacked parallel to each other. Varied number of cisternae are present in Golgi complex. The Golgi cisternae are concentrically arranged near the nucleus with distinct convex cis or the forming phase and concave trans or the maturing phase. The cis and trans phase of the organelle are entirely different but interconnected. The Golgi apparatus principally perform the function of packaging materials to be delivered either to the intracellular target or secreted outside the cell. Materials to be packaged in the form of vesicles from the ER fuse with the cis phase of the Golgi apparatus and move towards the maturing phase. This explains why the Golgi apparatus remains in the close association with the endoplasmic reticulum. A number of proteins synthesized by ribosome on the endoplasmic reticulum are modified in the cisternae of the Golgi apparatus before they are released from its trans phase. Golgi apparatus is the important site of the formation of the glycoprotein and glycolipids. Now lysosome. These are membrane-bound vesicular structures formed by the process of packaging in the Golgi apparatus. The isolated lysosomal vesicles have been found to be very rich in almost all the types of hydrolytic enzymes, hydrolases, lipases, proteases, carbohydrates. Optimally active at the acidic pH, these enzymes are capable of digesting carbohydrates, protein, lipids, and nucleic acids. Vacuoles. The vacuoles is the membrane-bound space found in the cytoplasm. It contains water-safe excretory product and other materials not useful for the cell. The vacuoles is found by a single membrane cell called tenoplast. In plant cell, the vacuoles can occupy up to 90% of the volume of the cell. In plants, the tonoplast facilitates the transport of a number of ions and other materials against concentration gradients into the vacuoles. Hence, their concentration is significantly higher in the vacuole than in the cytoplasm. In amoeba, the contractile vacuole is the most important for the osmoregulation and excretion. In many cells, as in protists, food vacuoles are formed by the engulfing the food particles. Mitochondria, unless specifically stained, are not easily visible under microscope. The number of mitochondria per cell is variable depending on the physiological activity of the cells. In terms of shapes and size also, considerable degree of variability is observed. Typically, it is sausage shaped or cylindrical, having a diameter 0.2 to 1 micrometer, average 0.5 micrometer, and length 1 to 4.1 micrometer. Each mitochondrion is a double membrane bound structures with the outer membrane and the inner membrane dividing its lumen distinctly into two aqueous compartments, that is the outer compartment and the inner compartment. The inner compartment is filled with the dense homogeneous substance called the matrix. The outer membrane forms the continuous limiting boundary of the organelle. The inner membrane forms a number of enfolding called cristae towards the matrix. The cristae increase the surface area. The two membranes have their own specific enzyme associated with the mitochondrial function. The mitochondria are the sites of the aerobic respiration. They produce cellular energy in the form of ATP. Hence, they are called powerhouse of the cells. Matrix also possessing single circular DNA molecules, a few RNA molecules, ribosome 70S, and the components required for the synthesis of proteins. The mitochondrial divides by fission. Plastids. Plastids are found in all plant cells and euglenoids. These are easily observed under the microscope as they are large. They bear some specific pigment, thus imparting specific colors to the plants. Based on the types of pigment, plastid can be classified into chloroplast, chromoplast, and then leucoplast. The chloroplast contains chlorophyll and carotenoid pigments which are responsible for trapping light energy essential for photosynthesis. In the chromoplast, fat-soluble carotenoid pigment like carotene, xanthophyll, and other are present. This gives a part of the plant a yellow, orange, or red color. The leucoplast are the colorless plastid of varied shape and size which stored nutrients. Amyloplast store carbohydrates, starch, example potato. Elioplast store oils and fats, whereas the eleuroplast store protein. Majority of the chloroplast of the green plants are found in the mesophyll cells of the leaves. These are lens-shaped, oval, spherical, discoid, or even ribbon-like organelles having variable length 5 to 10 micrometer and width 2 to 4 micrometer. Their number varies from 1 per cell of the Chlamydomonas, a green alga, to 20 to 40% per cell in the mesophyll. Like mitochondria, the chloroplasts are also double membrane bound. Of the two, the inner chloroplast membrane is relatively less permeable. The space limited by the inner membrane of the chloroplast is called the stroma. A number of organized flattened membranous sacs called the thylakoids are present in the stroma. Thylakoids are arranged in a stack like the piles of coins called grana, singular granum, or the integral thylakoid. In addition, there are the flat membranous tubules called the stroma laminae connecting to the thylakoid of the different grana. The membrane of the thylakoid encloses a space called lumen. The lumen of the chloroplast contains enzyme required for the synthesis of carbohydrates and proteins. It also contains small double-stranded circular DNA molecules and ribosomes. Chlorophyll pigments are present in the thylakoid. The ribosomes of the chloroplast are smaller, 70s, than the cytoplasmic ribosomes, that is 80s. Now, ribosomes. Ribosomes are the granular structures first observed under the electron microscope as dense particles by George Palade, 1953. They are composed of ribonucleic acid and proteins and are not surrounded by any membrane. The eukaryotic ribosomes are 80s while the prokaryotic ribosomes are 70s. Each ribosome has two subunits, larger and smaller subunits. The two subunits of the 80s ribosomes are 60s and 40s, while that of 70s are 50s and 30s. Here the S is Swedberg's unit, stands for the sedimentation coefficient. It is indirectly a measure of density and size. Both 70s and the 80s ribosomes are composed of two subunits. Now, cytoskeleton, an elaborate network of filamentation proteinaceous structures consisting of microtubules, microfilaments, and intermediate filament present in the cytoplasm is collectively referred to as the cytoskeleton. The cytoskeleton in a cell are involved in many functions such as mechanical support, motility, maintenance of shape of the cell. Cilia and flagella. Cilia and flagella are hair-like outer growth of the cell membrane. Cilia are the small structures which work like oars, causing movement of the either of the cell or the surrounding fluids. Flagella are comparatively longer and responsible for cell movement. The prokaryotic bacteria also possess flagella, but these are structurally different from that of the eukaryotic flagella. The electron microscopic study of a cilium or the flagellum show that they are covered with plasma membrane. Their core, called the exoneme, they possess a number of microtubules running parallel to the long axis. The exoneme usually has 9 doublet of radially arranged peripheral microtubule and a pair of centrally located microtubules. Such an arrangement of exonemal microtubules is referred to as the 9+2 array. The central tubules are connected by the bridge and each and is also enclosed by a central sheath, which is connected to one of the tubule of each peripheral doublet by a radial spoke. Thus, there are 9 radial spokes. The peripheral doublets are also interconnected by linker. Both the cilium and flagella emerges from centriole-like structures called the basal bodies. Centrosome and centrioles. Centrosome is an organelle usually containing two cylindrical structures and called centrioles. They are surrounded by amorphous pericentriolar materials. Both the centrioles in centrosome lie perpendicular to each other, in which each has an organization like the cartwheel. They are made up of 9 evenly spaced peripheral fibrils of the tubulin protein. Each of the peripheral fibrils is a triplet. The adjacent triplets are also linked. The central part of the proximal region of the centriole is also proteinaceous and called the hub, which is connected with tubules of the peripheral triplet by radial spoke made of protein. The centrioles form the basal body of cilia and flagella, the spindle fibers that give rise to a spindle apparatus during cell division in animal cells. Nucleus. Nucleus as a cell organelle was first described by Robert Brown as clearly as 1831. Later, the material of the nucleus stained by the basic dyes, which was given the name chromatin by Fleming. The interface nucleus of a cell when it is not dividing, has highly extended and elaborate nucleoprotein fiber called chromatin, nuclear matrix, and one or more spherical bodies called nucleoli, singular nucleolus. Electron microscopy has revealed that nuclear envelope, which consists of two parallel membranes with a space between 10 to 15 nanometer called perinuclear space, forms a barrier between the material present inside the nucleus and that of the cytoplasm. The outer membrane usually remains continuous with the endoplasmic reticulum and also bears ribosomes on it. At the number of the places, the nuclear envelope is interrupted by minute pores which are formed by the fusion of its two membranes. These nuclear spores are the passage through the which movement of RNA and protein molecules takes place in both directions between the nucleus and the cytoplasm. Normally, there is only one nucleus per cell. Variation in the number of nuclei are also frequently observed. So, can you recollect names of organisms that have more than one nucleus per cells? Some mature cells even lack nucleus. Example, erythrocytes of many mammals and sieve tube cells of vascular plants. Would you consider these cells as living? So the nuclear matrix of the nucleoplasm contains nucleolus and chromatin. The nucleoli are spherical structures present in the nucleoplasm. The content of the nucleolus is continuous with the rest of the nucleoplasm as it is not a membrane bound structure. It is the site for the active ribosomal RNA synthesis. Larger and more numerous nucleoli are present in the cell, actively carrying out protein synthesis. You may recall that interphase nucleus has a loose and indistinct network of nucleoprotein fibers called chromatin, but during different stages of cell division, the cell showed structure chromosomes in place of the nucleus. Chromatin contains DNA and some basic proteins called histones, some non-histone proteins, and also RNA. A single human has approximately 2-meter long thread DNA distributed among its 23 pair chromosomes. You will study the details of DNA packaging in the form of chromosomes in class 12th. Every chromosome visible only in dividing cells essentially has a primary constriction or the centromere on the sides of which disc-shaped structures called kinetochores are present. Centromere holds two chromatids of the chromosome. Based on the position of the centromere, chromosomes can be classified into four types. The metacentric chromosomes has middle centromere forming two equal arms of the chromosomes, and the submetacentric chromosomes has the centromere slightly away from the middle of the chromosomes, resulting into one shorter arm and one longer arm. In acrocentric chromosomes, the centromere is situated close to its end, forming one extremely short and one very long, whereas the telocentric centromere has a terminal centromere. Sometimes a few chromosomes have non-extending secondary constriction at the constant location. This gives the appearance of a small fragments called satellite. Microbodies. Many membrane-bound minute vesicles called microbodies that contain various enzymes are present in the both plants and animal cells. So that's it. Thank you so much.